Bismillah ar-Rahman ar-Rahim. Today we will continue our fourth lecture in neurophysiology. Remember that most neuronal cells consist of cell body or the soma and dendrites, that they are branching everywhere. Then there is this area that's called the axon hillock, the axon, and then the axon terminals. And we know from previous lectures that the action potential will start, or it will be transmitted to this neuron from the other neurons, or what we call it presynaptic neuron, and most of the synapses are chemical synapses. So this is a postsynaptic neuron, and we said that in chemical synapses the way the conduction is unidirectional. So it's always from the presynaptic neuron to the postsynaptic neuron. It comes through the synapses, either through the dendrites, the soma, or maybe the axon. Then it will propagate all the way through the axon into the axon terminals, and now the signal will be transmitted again in the synapses with dendrites or cell bodies of the postsynaptic neurons here in this case. Remember also that the action potential, usually these signals are coming to the neuron, will be as postsynaptic potentials that can be either excitatory or inhibitory postsynaptic potential, and they will be summated, but the generation of the action potential actually takes place here at the axon hillock. That's why it's called an integrative zone, whereas the input zone, which are the dendrites and the soma, they are the parts that receive the inputs from the other neurons. The signals are integrated here at the axon hillock, in which the action potential is generated, and then the conduction will be propagated all through the axons until the axon terminals. The change in the membrane potential at the level of the dendrites and the soma, it's usually in the form of postsynaptic potential, which is graded potential. It's not an action potential. It's very unlikely to result in an action potential. Whereas the generation, as we said, the action potential is usually at the level of the axon hillock, and then it will be transmitted throughout the axon and the axon terminal. The reason why the generation is usually at the level of the axon hillock, that as you see here, the highest concentration of voltage-gated channels, especially we're talking about voltage-gated sodium channel, that will do the depolarization phase of the action potential. It's actually concentrated here at this level, where we have very few of these voltage-gated channels in the dendrites and in the soma. But most of the channels here, they are ligand-gated channels or chemically gated channels. So when they bind to transmitters from the synapses from other neurons, they're going to change the permeability, allowing movement of ions, can be either excitatory or inhibitory postsynaptic potentials. And then this grand postsynaptic potential, the summation of them all will be reach the axon hillock, in which it has the highest concentration, as we said, voltage-gated channels. Then once the action potential is generated, it will be transmitted here. In this case, this is a myelinated nerve. So this neuron is covered, the axon is covered with myelin sheath. So you can see here that it's a saltatory kind of conduction of the nerve impulse to increase the speed of conduction. Then the action potential will reach to the axon terminals, and then the neurotransmitters will be released. So this is where we have action potential. This is a graded type of potential, or a postsynaptic potential. When we talk about neuronal synapses, then which parts exactly of the neuron are going to synapse with each other? Always we're going to start with the axon terminal, as we said. So if we want to name this kind of synapse, which all are we talking about are chemical synapses. So because it starts with the axon, we call it axo, and synapsing with the dendrites, so it's called axodendritic synapse. And this is the most common type of synapses. The other type of synapse, that the axon, we're going to do the synapse on the cell body. So this is axosomatic synapse. The third type of synapses, it could be on the axons anywhere. So we call it axonal synapse, like in this case. But as we said, the most common type of synapses here are the axodendritic synapses. Let's start talking about the axodendritic synapse. So you can see here from the dendrite that they are branching everywhere, so the spatial field is very large. There is higher opportunity for increasing number of synapses, and then a greater opportunity for the summation. For example, you can see a synapse here, here, anywhere here in the dendrites we can have synapses, and then increasing the number of synapses, increasing the opportunity of firing and action potential if they are excitatory, or maybe canceling each other if some are excitatory and some are inhibitory. So again, this is the axodendritic synapse. Dendrites are characterized by having what we call it decremental conduction, which means the change in the membrane potential will decrease, the efficacy will decrease as we are going toward the soma, which again, in other words, means that the same synapse will be more effective if it's nearer to the soma than if it's further away from the soma. So again, it depends on the distance. The explanation for this decremental conduction in the dendrites is that the dendrites are long and their membranes are thin. On the membranes, what characterizes the membranes here in the dendrites, they are leaky to potassium. And you know that the potassium will act oppositely to the effect of the sodium. Sodium will cause depolarization, whereas potassium will cause repolarization, especially that the equilibrium potential is near the resting membrane potential. So if there is, for example, an excitatory signal here, let's say this is E1, is about like 10 millivolt, then the change in the membrane potential, for example, let's say this E1 equals 10 millivolts. And excitatory, it will have a positive sign because it's in the toward the positive direction, or it's a depolarizing kind of thing. And let's say that the resting state in this neuron is minus 70 millivolt, and the threshold here is minus 50 millivolt. So let's say this excitatory signal is plus 10 millivolt. So that means that the membrane potential will change from resting membrane potential, which is minus 70, into minus 60 here. But that doesn't mean that the membrane potential here in the soma will be minus 60. It's very unlikely to be there, because here there is minus 60, then leakage of the potassium will cause it to be, let's say, minus 65, then again minus 70. So the conduction is decremental. The strength of the excitatory signals will decrease as we are going toward the soma. However, if this excitatory signal was here, the effects could be higher. It depends also on the other signals that coming all the way here, whether they are excitatory, inhibitory, and what is the value of each one of them. Let's take this example. If we assume in this neuron there is an inhibitory signal minus 5 millivolt, whereas the excitatory signal here is about 10 millivolt. Assume that the resting membrane potential in this neuron is minus 70 millivolt. So calculate this grand postsynaptic potential in this neuron, assuming that they are only the two synapses are coming now. How about the axosomatic synapses? See here in the soma, it's characterized by having a relatively large diameter, which means less resistance to conductance. The fluid here in the soma is highly conductive, so any change in the membrane potential will be conducted or will be the same anywhere in the soma. For example, here a synapse with the plus 10 millivolt will change this membrane potential from minus 70 into minus 60 here, but this is the same. It will be minus 60 here and it will be minus 60 here, unless there are other synapses. Then will we calculate the summation of these synapses. Assume this resting membrane potential in this neuron is minus 70 millivolt. This is an excitatory synapse with the value of 10 millivolt. Calculate the grand postsynaptic potential in this neuron. Assume in this neuron, the same neuron, which has a resting membrane potential of minus 70 millivolt, and we have this E1 of plus 10 millivolt. Let's say we have another excitatory signal here in this case, which is E2. Again, it's a plus 10 millivolt. It's not very realistic actually, because one synapse is most likely to change the membrane potential of 0.5 to 1 millivolt. Not that much. Usually we need so many synapses to reach this level. And let's assume that the threshold here is minus 50. So do you expect this neuron, if we are assuming that these are the only synapses at this time, does this neuron, is it gonna reach the, is it gonna fire action potential or not? But just one note, that when we say action potential propagation, we may say fire, or we may say discharge, or in this example, this neuron has a resting membrane potential of minus 70 millivolt. It has an excitatory signal E1, 10 millivolt, another excitatory signal E2, 10 millivolt, and an inhibitory signal I1, it's a 20 millivolt. Threshold is 50 millivolt minus 50 millivolt. Calculate the grand postsynaptic potential in this neuron, assuming that these are three synapses that fire simultaneously, or they act simultaneously on the soma of this neuron. So again, calculate the grand postsynaptic potential, and then determine if there is gonna be a firing of action potential or not, or a generation of action potential or not. This example shows the complexity of the synapses of different neurons on the same postsynaptic neuron. You can see how they will gonna change the membrane, especially if we're talking about the exodendritic synapses. You can appreciate this decremental conduction in these synapses, and you can see the effect of the excitatory signal versus the inhibitory signal and how they gonna change the membrane potential. Also, you can appreciate the powerful effect on these synapses. They are axonal synapses, and they are more specifically acting almost on the axon helix. Another common and very important type of axonal synapses, it's called the presynaptic. Either inhibition or facilitation depends on the change in the membrane potential that's gonna happen. So assume this is the synapse of interest, and let's say it's an excitatory synapse. So an action potential is gonna come here to the axon terminal. Then it will activate a voltage gated calcium channel that will help to activate the process of exocytosis. The neurotransmitter will be released and then attached to the postsynaptic receptor, let's say an ionotropic receptor. So it's going to change the membrane permeability. For example, is an excitatory signal. So, and again, it will fire an action potential. Actually, sometimes in the nervous system, in the central nervous system, we want this pathway to continue, but we want to modulate this type of pathway every now and then, depending on the stimulus, depending on the sensory impulses that we're going to receive. Or maybe this is an important mechanism in learning and memory. For example, for learning and memory, this pathway is going to continue. But in this instance, when this pathway comes, let's say I don't want to remember this, or I don't want this pathway to continue. I'm going to stop it here. So what happened here, that this neuron is going to send a neurotransmitter that's an inhibitory neurotransmitter. Usually it's GABA that will bind to chloride receptors and cause hyperpolarization. So it will decrease the change in the membrane potential, or it will alter the change in the membrane potential. So the action potential that's supposed here to come and open the voltage gated calcium channel, it may be no longer able to open this calcium channel, and therefore the release of the neurotransmitter. So we're going to inhibit this action of the synapse. That's why it's called presynaptic inhibition. Sometimes we want the opposite. So in this kind of presynapse, the neurotransmitter receptor complex is excitatory. So you're going to even increase the potency of this synapse. So we call it presynaptic facilitation. So you're going to alter the activity of this synapse depending on the other factors that are controlling this thing. One of these important applications of the presynaptic inhibition is in the sensory system. We have what we call it lateral inhibition or a presynaptic inhibition. For example, in vision, to make the border of the image is much more clearer. In this example, assume that the resting membrane potential in this neuron is minus 65 millivolt. Threshold is minus 55. We have an excitatory signal coming here as 10 millivolt, another excitatory signal of 5 millivolt, an inhibitory signal of 20 millivolt. These are the only three signals affecting this neuron at this specific time, and they are affecting the soma simultaneously. Calculate the grand postsynaptic potential, or the summation of these postsynaptic potentials and determine if there will be a discharge or the generation of action potential or not in this case. This graph shows that neurons in our nervous system, they have different characteristics. Although they all share the same main principle, which is what we talked about, the action potential, postsynaptic potential, the chemical synapses, the neurotransmitter receptor complex, all these kind of things. But neurons, they have different characteristics because they serve different functions. Sometimes I need this neuron to transmit this signal super fast. Sometimes it's not that necessary to transmit it very fast, so they differ in their speed of conduction. Sometimes I need these types of neurons to fire almost continuously, so they will have very low threshold for generating action potential. Sometimes this neuron is better to send more frequent action potentials, to have the ability to increase the frequency of discharging these action potentials. For example, in this neuron, you can see the red one is neuron 1, the blue is neuron 3, and green is neuron 2. You can see that neuron 1 here has almost the lowest threshold, so it fires very fast. But you can see that the maximum frequency of discharge is for neuron 3, which has almost the highest threshold. So it takes long until it generates the action potential, but once it's reached, it can reach a maximum frequency of action potentials per second that is much higher than neuron 1 or even neuron 2. So again, these kinds of differences in their exact value of the threshold for the action potential, in their frequency of discharge or the rate of discharge in each neuron, they are very important and they serve different functions because the nervous system is composed of different pools, and these pools will serve different functions. And as you know from histology, that neurons can be either myelinated or unmyelinated. They even differ in their diameter. We have a wide range of changes in diameter or between neurons. Like they range from 0.5 micrometer into 20 micrometers. And you know also that this kind of increase in the diameter in these neurons is very important to increase the conduction velocity, to increase the speed of conduction of the nerve signals among these neurons. Also, the myelinated nerve fibers are very much quicker or faster in transmitting signals than the unmyelinated one. And you know there are several classifications for these nerve fibers or neurons depending on their diameter and also the myelination. For example, nerve fibers type A are much faster and higher conduction velocity than nerve fibers C. C are small fibers, they are unmyelinated. So if you have a signal that you want this signal to be transmitted super fast, would you choose fiber A or fiber C? It's definitely fiber A. For example, in our reflexes, like stretch reflexes in the spinal cord, you want this signal to reach very fast and finish this reflex super fast in order to protect the muscle from injury, to maintain the balance of the body. So these type of signals, they have A fiber, and it's very fast. Whereas if you're talking about, for example, the itch sensation, the tickle type of sensation, they are transmitted through type C fibers because they are not critical to the function of our body. Neuronal cells, they are grouped together in different groups, so they're going to serve different functions in the nervous system, and they have different ways of communication. For example, here we have an input neuron 1 that's synapsing with postsynaptic neuron A, and sends also branches to B and C. And we have this neuron 2, it's also sending synapses to C, you can see also to B, and also to D here. So this the area in which this neuron will act on the postsynaptic neuron, for example here, it's called this stimulatory field of this neuron. If we are assuming that the effect is excitatory from this neuron to the postsynaptic neuron, so we call this area, the effect of this neuron, is stimulatory field. This is stimulatory field of neuron 1, and this is the stimulatory field of neuron 2. And you can see here in this stimulatory field that the concentration of the synapses is the highest in the center. So if we say there are more synapses here in this area, and this is an excitatory signal, so there is a higher chance of reaching a threshold, a higher chance of discharge. So that's why this central area of the highest concentration of synapses is called the discharge zone. Whereas on the periphery, this kind of excitatory signal is not strong enough, and the number is not that large enough to reach a threshold. So what it causes what we call it facilitation. So it's making nearer to the threshold. It makes it easier. It facilitates that in the next time or by the next synapse, this neuron may fire an action potential. So these are the discharge zone in the center and the facilitation zones in the periphery. If we assume that neuron 1 is an inhibitory neuron, the effect of the neurotransmitter to receptor complex is inhibitory in this case, then this field or this area is called an inhibitory zone of a neuron 1. Okay, and again the greatest inhibition will be at the center of this area of the inhibition, because of the highest number of synapses, so that's the highest effect. Neurons are connected in different ways with each other, so they can perform different functions and modulate different kind of functions. These different types of connections, we call them neuronal circuits. Let's take examples on common neuronal circuits in the nervous system. In this case, this kind of neuronal circuit, we call it a reflex, and this is a spinal reflex because the integration is the level of the spinal cord, and we call this reflex is a stretch reflex because the stimulus here was a stretch stimulus. So in this case, this is a very common reflex that we examine during neurological examination in the clinic. So you're going to ask the patient to sit and relax. Then you're going to hit the patellar tendon with the hammer. So what you're doing here, you're stimulating this muscle, you're stretching this muscle. So that will activate specific receptors here in the bulk of the muscle. It's called stretch receptors or the muscle spindle. That signal will be sent through the sensory neuron, the one in blue, and it will enter the central nervous system to the spinal cord. And it will synapse immediately with a motor neuron. and this motor neuron will act to stimulate this muscle to contract. So you are stretching the muscle. The proper response is for this muscle to contract to protect this muscle fibers from injury. This is the shortest stretch reflex in our body. It's called monosynaptic. So you can see in the stretch there is only one synapse. So it's a monosynaptic reflex. It results in the stimulus is a stretching of the muscle, and the response is contraction of the same muscle to protect this muscle. This is a monosynaptic reflex. And as you see here, that this sensory neuron is sending other branches. Remember when you learned about the limbs, the movements of the limbs, that we have antagonistic muscles in our limbs. So if there is a muscle that will do an extension on the knee, there is another muscle that will do flexion on the knee. So if you are contracting a muscle, you should relax the antagonistic muscle. And this is the same in this case. So if I'm contracting this extensor muscle, I have to relax the flexor. But how do we do this if we have the same stimulus, the same sensory neuron, which has almost the same sensory, or it has the same neurotransmitter? In this case, this is an excitatory neurotransmitter. So the thing here that if you want to do an excitatory effect on a side, you're going to do an inhibitory effect on the other side. So in this case, we added here an interneuron, and this interneuron is an inhibitory neuron. So you can see here this sensory neuron sending excitatory signal to this interneuron, and this interneuron is synapsing with the motor neuron of the antagonistic muscle. This inhibitory interneuron releases an inhibitory neurotransmitter, for example, glycine, and that will cause inhibition of this motor neuron to the skeletal muscle, the antagonistic skeletal muscle, causing inhibition in this muscle. Because this is an opposite effect, this is a reciprocal effect, we call this circuit reciprocal inhibition. So in reciprocal inhibition, this is the same signal that's coming here. It's causing excitation in one direction, but it's causing inhibition in the other direction. And this is very especially important in the controlling of the antagonistic muscles of the limbs. You need to do excitation of the muscle, but inhibition in the antagonistic muscle, and vice versa. So this is reciprocal inhibition. Note here that this sensory neuron, this neuron branches to give different stimuli to different outputs. For example, this first branch to stimulate the muscle, the other one to inhibit the antagonistic muscle, and this is the third one to give an information to the brain, to the higher areas in the brain about this change during this reflex. You need to be consciously aware of that. You need to know this kind of information in order to coordinate your balance, to do coordination. So this divergence of signal is important to serve different functions in the nervous system. Generally, the divergence circuits are divided into divergence in the same tract or divergence into multiple tracts. And as we mentioned, that divergence into multiple tracts is very important to have this holistic kind of integration of information to this integrative part of processing information. For example, when you move your head, there are specialized receptors, sensory receptors present in the inner ear. This kind of information will be transmitted through one of the cranial nerves called the vestibulocochlear nerve. This nerve will enter to the nuclei in the brainstem. This vestibular nuclei has to diverge. This signal from the vestibular nerve has to diverge into multiple tracts. For example, when you move your head, you need to move your neck. You need also to maintain the balance in your body by making proper contraction relaxation of your axial body muscles. You also need to coordinate that with movements in your eyes, so you can fixate that on special visual field so you don't fall down. Also, you need to send these signals to the cerebral cortex so you can consciously aware of the movement of your head. This is an example of divergence into multiple tracts. Divergence in the same tract is important to serve a function of amplification of the signal, amplifying this signal. For example, in the motor neurons, which are the neurons that gonna supply the skeletal muscle fibers, can see if there is one motor neuron for each muscle fiber, we have to have like thousands to millions of motor neurons, which is not efficient. So the other way is that for this same motor neuron to branch several times to diverge into multiple muscle fiber cells. So these muscle fiber cells, they act as a one unit. It's called the motor unit to contract at the same time. So that give you a more pronounced type of movement or more effective movement. So again, this is an amplifying divergence example. The nervous system sometimes needs convergence rather than divergence. And convergence, you get multiple signals to unite on one output or a single neuron. And this is important. Can be also from a single source, or it can be from separate sources. They are important for summation, for magnifying these kind of effects. An important example of convergence is what happens at the level of the retina in our eyes. We have photoreceptors, they are called rods. And rods, they help us see in the dark. So it's very important for us in the dark to detect even the tiny changes in the light or the tiny stimuli from the light. So these rods act like this so we can see better in the dark. And what helps with that is the convergence on a single ganglionic cell or a neuron that's called a ganglionic cell. Because, like, for example, this excitatory or this postsynaptic potential that's occurring at the level of the receptor, it's most likely it's not enough to cause a firing in the action potential to reach a threshold. Let's say here a change in each one of them will change the membrane potential by 1 millivolt. However, to reach a threshold, you need a change of about 5 millivolts. And so 1 millivolt here will not be reached here or will not be translated into an action potential and reach the cerebral cortex. So you can be aware of this light. However, if all the 5 rods together, they converge on the same postsynaptic neuron, and then now a change of 5 millivolt is enough to reach a threshold, then that means it reaches the cerebral cortex, and we can be consciously aware of this change in the light, so we can see these tiny changes in the light during the dark period. Sometimes the nervous system needs for a signal to be prolonged, to have a prolonged effect. And we talked previously when we talked about synapses that we can do prolongation of the effect if the neurotransmitter act on the metabotropic receptors rather than an ionotropic receptors. So that will cause a longer action on the postsynaptic neuron. And another example of prolongation signal is something we call it parallel after discharge. And it's simply that if we're talking about this neuron as an first neuron, it's firing, so it's sending the signal through this synapse with the second neuron. So signals from the input neuron, the blue one, to the orange neuron, the output one, okay? But if we need this signal reaching this orange neuron to be prolonged, I need this neuron to fire for a longer time. But this signal is coming from this input neuron, the blue one. It's gonna last for a few milliseconds, then it's gonna stop. So one of the mechanisms that the nervous system does is that we do branches collateral of the same neuron that's firing, and what characterize these branches that they're gonna synapse with different numbers of neurons. So again, this is a direct synapse with this orange neuron, but here there is two interneurons in between. And here there are three interneurons in between. What that means? That you know that the principle of delayed transmission at the level of the synapse. So if you are increasing the number of synapsing, you are delaying this effect or this signal more and more. We talked that, for example, let's say that this transmission through this neuron took 1 millisecond, and then the delay at the level of the synapse 0.5 millisecond. So this signal will reach this orange neuron in 1.5 milliseconds. If we add here this 0.5 millisecond in the synapse, another 0.5 millisecond here, then we add the distance, also increase in distance. So the same type of stimulus that was fired from this neuron will reach here, let's say, in 2 milliseconds, but the neuron will fire an action potential. In another 1 millisecond, then the same signal will reach, so another action potential. Then in a 3 milliseconds, this one. So we are outside the refractory period, but we can fire this sequence of action potentials from this neuron. So we are prolonging the discharge or the signal coming from this pathway. Please be sure that you do not confuse it with what we will explain next as a reverberatory circuit. Another example of prolonging the signal transmission is what we call it reverberatory circuits. In this case, this signal action potential is transmitted from the input neuron to this output neuron, which will fire now an action potential. But let's say that the nervous system wants this neuron to almost continuously or to prolong this output from this neuron. So in this case, there is a collateral, there is a branch coming out from this same neuron, and again it's synapsing with the same neuron. Like, see see there is a collateral coming out from this neuron, and then the axon terminal, in this case, synapsing with either the dendrites or the soma of the same neuron. So again, another action potential will continue, and that will continue until something happened in the nervous system, and we will talk about this later, whether this an inhibitory signal or a fatigue happen at this type of synapse. Again, this reverberatory circuit, it's like a positive feedback circuit, okay? It's different from the parallel after discharge circuit. This figure is to show you that the reverberatory circuits can get much more complex. This example to show that these reverberatory signals, we need them for almost a continuous type of signaling that we need it all the time. But circumstances can happen and changes in the environment can happen that we need to modulate the rate of the signaling in this pathway or changes in this pathway, whether either we do facilitation of this pathway or inhibition of this pathway. For example, let's say this kind of reverberatory circuit controls the respiratory rate in our body. So you need to breathe all the time. So you need these signals all the time coming. But sometimes, let's say you are exercising, so you want to increase your respiratory rate. So you're going to activate the facilitation kind of circuit, increase the respiratory rate. You need to breathe more to get more oxygen for your muscles to exercise. But sometimes you need to inhibit or decrease this respiratory rate. Different areas of the nervous system are connected to each other in a way or another. So it's very important to maintain stability of the neuronal circuit. You don't want to be a continuous excitation from all neuronal circuits all the time. So there are two important mechanisms to maintain this stability of the neuronal circuits. The first thing, inhibitory circuits. So these circuits can be like what we call it the basal nuclei. We have specialized structures in our brain. They are called basal nuclei, and they send inhibitory signals to the cerebral cortex, which the motor cortex that control our skeletal muscle. So in case of impairment in the function of these basal nuclei, one of the manifestations that will cause rigidity in the muscle. So you will no longer, because of this excess stimulation in the muscles, it will interfere with your daily activity, with everything activity, because this is abnormal to be excited all the time. The other mechanism of stabilizing the nervous system is the fatigue. And fatigue can occur at different levels, especially at the synapse. And one of these mechanisms could be there is depletion of a neurotransmitter. We have no more neurotransmitter. Maybe changes in the membrane potential postsynaptic neuron that it's not responsive yet, or saturation of the receptors in the postsynaptic neuron. And fatigue is, for example, it's very important in patients with epilepsy. You know, those patients when they have, especially the generalized type of epilepsy, when they have seizures attacks, they kind of have these tonic-clonic attacks for seconds or maybe minutes, but then they stop. And the reason for that is the fatigue that happens in the nervous system because of the depletion of these neurotransmitters allow them to relax and to stop these kind of attack, which is a protective mechanism, as we said. Neuronal excitability and synaptic transmission can be affected by external factors, such as changes in the pH, in the acidity of the fluids inside our body. For example, in cases of alkalosis, like hyperventilation or maybe a panic attack when you wash out CO2 out of your body, it's dangerous on patients with epilepsy, because this kind of alkalosis will increase the neuronal excitability and that will induce more seizure attacks in these patients. That's why we advise them to try to control their panic attack and learn different techniques to control it. On the other hand, acidosis leads to depression in the neuronal excitability on the activity. For example, in patients with diabetes, when they have severe diabetic ketoacidosis, those patients develop coma, actually decrease the level of consciousness. Neuronal cells are especially sensitive to changes in the level of oxygen. So, if there is any hypoxia or a decrease in the oxygen supply to the neuronal cells, even if that's like for 4 or 5 seconds, that may lead to development changes in the level of consciousness, and these patients may develop coma. Food, drink, supplements, drugs, they also can interfere with the excitability of the neurons. And common examples are caffeine in coffee, theophylline in the tea, and theobromine in the cocoa. They all can increase the excitability of the neurons. Whereas, for example, the anesthetic drugs will decrease the excitability of the neurons. Have a look at these examples and determine what type of neuronal circuits are they. And now we are done with our lectures in Introduction to Neurophysiology. Next time we will dig in details in neurophysiology when you take the nervous system during third year medical school. Good luck and thank you.